Hello, everybody. My name is Dr. Hannah Smith, and today I'm going to be presenting the presentation that I gave for MND Scotland's LEARN events at both Glasgow and Edinburgh. I'm a postdoctoral researcher with the University of Edinburgh, and I'll be talking today about my project, which is profiling the actively synthesized proteins in motor neurons during different stages of a model of motor neuron disease. So thanks for listening. So I'll just introduce myself and my current research just so that I'm not a stranger on a screen. So I earned my PhD in neuroscience in 2019, and now I'm what you would call a postdoctoral research fellow because I have already got my doctorate at the University of Edinburgh. I'm what you would call a fundamental or preclinical researcher. So that means I don't deal with patients or people on a daily basis. I'm at the lab bench and I'm looking into really the foundational biology of motor neurons so that we can better understand motor neuron disease. The research that I'm going to be talking about today was funded by MND Scotland. And I'm very proud and honoured to say that I am the first early career researcher member of MND Scotland's Scientific Advisory Panel, what we call the SAP. And the SAP is a panel that reviews and directs which research is funded by MND Scotland. So if you go onto their website at the moment, you'll see my bio as well, with a little bit of information about me. So when we're thinking about motor neuron disease, both from a researcher perspective and from the public perspective, there's several questions that we can ask. So a big problem that a lot of people know is that there's no definitive test for motor neuron disease, which is why we have diagnosis delays and other sort of issues with finding out that people actually have MND. And if I rephrase this question as a scientist, I could rephrase it as, we need to find new proteins present in MND that we can use to make diagnostic tests with that we don't have at the moment. And a second problem we could say is that, as we all know, there are almost no effective treatments for motor neuron disease. And again, if I rephrase this as a scientist, I would say we need to find new proteins that can be targeted by drugs for treating MND. And the thing about these two questions is that they really have kind of a common question that we can ask, which is at early stages of motor neuron disease, what changes are actually taking place in the motor neurons that makes them unhealthy? And specifically for me, I'm focusing on what proteins are different between healthy and MND motor neurons that we can first identify and then target with something like a new treatment. So I'm going to just chat a bit about protein synthesis and why it's important. So the cells in our body are highly dynamic. They're constantly changing and reacting. They have to react to the changing environment around them. And they also have to perform their function according to what type of cell they are. So obviously a liver cell has a certain set of functions it needs to do. A heart cell has a certain amount of functions it needs to do. And a motor neuron has a certain amount of functions that it needs to do. And to do this, the cells are constantly producing proteins that are specific to the type of cell they are and what the cell needs. So for example, a motor neuron cell doesn't need to produce a lot of proteins that would be useful for a liver cell to make. And a liver cell doesn't need to make the same amount of protein that a heart cell needs. And obviously we know motor neurons are incredibly complex. They do a very, very difficult job in our body. And so their protein synthesis must be very, very delicately balanced. And this can go wrong in motor neuron disease. So if we zoom in, we see these tiny little protein synthesizing factories sort of working away constantly in our motor neurons, but also constantly in all the cells of our body. So we do know that protein synthesis is impacted in motor neuron disease. And I'm focusing on a certain protein called TDP43. So this is a protein that normally stays inside the nucleus of motor neurons, and it has an important function in healthy cells. So in a healthy motor neuron, the TDP stays clustered in this little spot. However, we do see from research that 95% of people with MND have TDP43 building up elsewhere in their motor neurons. So it leaves the spot where it's meant to be and kind of spreads around. And this is not good. It's not doing the right job for its role. And specifically for our purposes, TDP43 does actually directly interact with the protein synthesis machinery in motor neurons, and it can disrupt their normal function. So in other words, by leaving the nucleus in MND, it's kind of preventing the normal protein synthesis from happening. But one of the unanswered questions that my research has been aiming to look into is which proteins are actually most affected by this disruption during protein synthesis, and how can we determine what they are? So I use a technique called translating ribosome affinity purification. And I really appreciate that's a very long, complicated name, but the way that I like to think about it and the way that most people should just think about it is trap. So in a healthy motor neuron, we have kind of tagged with a little flag the protein synthesis machinery, and we've done the same thing for an MND motor neurons. And you can see that in this diagram, the little red one's TDP that's kind of disrupted it. And the trap basically is exactly that. It's a technique where we can trap the proteins that are actively being produced by this synthesis machinery in motor neurons at different disease stages. And the idea is that we build snapshots of what's being produced. And we can do this between healthy and diseased motor neurons and at different time points according to how advanced the disease is progressing. And I like to think of this as a spot the difference map. So imagine that each of these images is just a different protein that we're kind of looking at, and I have to pick through and find out what's really different. And for example, the proteins that are present only in the motor neuron disease data, so they're not in the healthy, we could think that these proteins might be toxic to motor neurons. And so a disease treatment would look at trying to reduce that amount of this protein. But on the other side, a protein that's only present in the healthy data and it's missing from the MND data, it might actually be protective in motor neurons, or it might be a really important protein that's required for motor neurons to be healthy. And so we would look at finding a treatment that could boost that level back up to normal, and hopefully that would improve the motor neurons' health in MND. So that's all well and good as kind of an idea of what the data looks like. But what does this data actually look like for me when I get it? It's actually just a gigantic Excel spreadsheet that's very complicated and has a lot of different aspects that we're looking at. And it's quite a lot to get through. After I've analysed all this data, here's a graphed example of what I found. So each of these dots is a single protein in the cell. And in total, we have profiled 19,117. And the color coding shows that gray has no difference between motor neuron disease data and healthy data. The red is reduced in MND and the blue is increased in MND. And it's just under 200 for each of these. And if we dig a little bit further in with this pie chart, 110 are reduced in the MND group, but they make functional proteins that have a role in actually doing the motor neuron function. And 82, the remaining, are reduced in the MND group. And they're not really functional proteins per se, but they're more a regulatory sort of pseudo function. And for those that are upregulated, 114 are increased in the MND group and they make functional proteins. And around 70 are increased but are non-functional and kind of, again, this regulatory group. So what's our next steps now that we have this data? So we have determined that 376 proteins are affected during protein synthesis in the early stages of our MND model, so right when symptoms are onsetting. What's interesting is that many of these proteins haven't actually been linked with MND before. And that's partly because investigating the actual protein synthesis machinery is a really cutting edge technique, this trap technique. So we haven't really had a clear picture previously of what's going on. And so this is really exciting for us because it throws up kind of a lot of potentially unknown or uninvestigated proteins that are actually really interesting. But of course, they do need more investigation. So, for example, what I'm looking at the moment is, are these proteins linked to a certain set of functions in the cell? For example, are the majority of them linked to how the cell gets its energy? Are they linked to how the cell actually performs its signaling function to make your muscles move? Is it linked to kind of structural? Is it that they're just weaker and so they get damaged easier? We don't know yet. Then the next point would be, are there already drugs available for these proteins that we can repurpose for MND, which is a big field at the moment, if we already know there are safe drugs that can be used for these new targets that we're coming up with. And also we need to kind of validate these findings in another model, for example, human cells or another animal model, just to see if really we can prove that this is a real change that we're seeing across kind of the spectrum of motor neuron disease. So I'd just like to say thank you very much for listening to me. Here are the names of the people who worked on this project alongside me. My supervisor, the head of my lab, is Professor Tom Gillingwater. We also collaborate with Trento in Italy and Utrecht in Netherlands. And obviously I said I was funded by MND Scotland and also under the University of Edinburgh. And if you want to find me at any of the MND Scotland events, the LEARN events, I have pink hair, so I'm quite easy to find, or you can follow me on Twitter at Dr. Hannah Smith. So thank you very much, and I hope you enjoyed my presentation.